We will start this module with a brief introduction to circulation and blood. The topics that will be covered here are blood, blood clotting, blood groups matching or compatibility, and blood transfusion. And at the end, we will summarize these topics through a brief recap. The human body is a wonder of nature. It is a complex organization. It is a complex organization of so many structures. Looking at these structures, many questions arise in our minds. For example, we know that all cells need oxygen for production of energy. But have you ever wondered, how do the deep-seated body cells get oxygen from the lungs? Have you pondered, how do the hormones produced in the brain find their way into all the cells? How does our body maintain a uniform internal environment in spite of constantly changing external environmental conditions? There is an organ system that takes care of all these functions. A finely regulated circulatory system in the human body, which ensures the transportation of important substances and also the maintenance of homeostasis. Components of circulatory system: blood, lymph, heart, and vessels. The blood vascular system may be of two types: open circulatory system and closed circulatory system. The open circulatory system is found in arthropods and mollusks. Closed circulatory system is found in vertebrates and a few invertebrates. In this form of circulatory systems, blood is closed within the vessels. These vessels are of different size and have varying thickness of the wall. Blood is pumped by the heart and travels through these vessels.
Let us look at the blood flow through the body in a little more detail. The oxygenated blood is pumped by the left ventricle into the aorta. Aorta now distributes the blood to all the organs through arteries. Veins bring back the deoxygenated blood from the tissues to the major veins. Superior vena cava drains the upper body, whereas the inferior vena cava drains the deoxygenated blood from the lower body. The deoxygenated blood in the heart is pumped into the lungs and comes back into the heart to begin the cycle once again. As the blood always flows inside the closed vessels and never enters any open space, it is termed as closed circulatory system.
Let us know more about blood. Blood is a fluid connective tissue comprising of a fluid matrix called plasma and formed elements. The following animation will explain the various components of blood.
Blood is a specialized connective tissue that consists of fluid matrix, plasma, and formed elements. If we centrifuge and try to separate the fluid matrix from the cellular components, we will find that nearly 55% of blood volume is plasma, while the rest 45% volume is taken up by the cellular components. Plasma is the liquid or the cellular component of the blood. More than 90% of this fluid matrix is water. It also contains dissolved proteins, glucose, mineral ions, hormones, carbon dioxide, etc., in addition to the platelets and blood cells. The plasma without the clotting protein, fibrinogen, is termed as serum. Cellular or formed component comprises of red blood cells, white blood cells and platelets. Erythrocytes or RBCs have a biconcave shape and are devoid of nucleus. This shape is significant as it increases surface area for gas exchange and allows the blood to flow easily through the narrow vessels. There are about 4.5 to 5.5 million RBCs present in 1 milliliter of blood. The red blood cells are synthesized in the bone marrows of ribs, sternum and vertebrae at the rate of 1.2 million cells per second. The lifespan of these cells is only about 120 days. Their main role is to carry oxygen to all the cells. Oxygen is bound to the hemoglobin present in the RBC. 95% of the cell protein is hemoglobin. The second cellular component is WBC or leukocytes. They are nucleated, have amoeboid shape, and play a significant role in the defense mechanisms of the body. The amoeboid nature of the leukocytes helps them to squeeze through the walls of the blood vessels in order to engulf bacteria. They do not contain hemoglobin. They number 6,000 to 8,000 per milliliter of blood. The ratio of RBC to WBC is nearly 1,000:1. There are 5 types of leukocytes which are categorized as granular and agranular types. Granular leukocytes are eosinophil, neutrophil and basophils. Eosinophil contains granules that take acid stain like eosin. Their nucleus is bilobed. They number 100 to 400 per milliliter of blood. Lifespan of eosinophil is 3 to 5 days. They play a very significant role in allergic responses. Neutrophil contains neutral staining granules. Their nucleus is 2 to 7 lobed. They number 3,000 to 6,000 per milliliter of blood. Lifespan of neutrophil varies from 12 hours to 3 days. They destroy foreign harmful organisms by ingesting them. This function occurs by phagocytosis or cell eating. Basophils contain granules that take basic stain. Their nucleus is 2 to 3 lobed. They number 25 to 200 cells per milliliter of blood. Their lifespan is 9 to 18 days. They develop into mast cell of connective tissue and secrete important chemicals that play an important role in mediating allergic response. Agranulocytes are of 2 types, monocytes and lymphocytes. Monocytes have a large kidney-shaped nucleus and abundant cytoplasm. They number 100 to 700 cells per milliliter of blood. They become macrophages in tissue upon stimulation and attack pathogenic bacteria. They also feed on dead tissues. Lymphocytes are agranulocytes that are produced in the lymphatic system. Their nucleus is large and occupies most of the cell. Their cytoplasm is basophilic. They have a lifespan of 100 to 200 days. They produce antibody on being exposed to antigenic molecule. The relative abundance of these cells are neutrophil 60 to 65%, eosinophil 2 to 3%, basophil 0.5 to 1%, monocytes 6 to 8%, lymphocytes 20 to 25%. The third cellular component of blood is platelet, which is responsible for blood clotting. Blood is.
Do you know that every second more than 2 million red blood cells get destroyed, but they're immediately replaced by new ones? After knowing about the components of blood, let us learn about its functions. Blood transports oxygen, carbon dioxide, hormone, waste, and nutrient to different organs and tissues. Blood regulates pH, temperature, and water balance in the body. Blood also protects us against foreign microbes. Blood cells help in blood clotting. After learning so many things, this makes athletes. After a cut or a bruise, you may have seen blood forming a clot. Do you know what is blood clotting and how a clot is formed? The process of changing of blood from fluid state to gel state is called blood clotting. There are two major facets of the clotting mechanism: platelets and thrombin system. The three steps of blood clotting are liberation of thromboplastin, conversion of prothrombin into thrombin, conversion of fibrinogen into fibrin.
The third cellular component is platelet, which is responsible for blood clotting. The damage to small blood vessels or capillaries frequently occurs due to external or internal reasons. When these blood vessels are damaged, there are three basic mechanisms that promote hemostasis or the stoppage of bleeding. Following the damage, there is an immediate reflex that promotes vasoconstriction, thus diminishing blood loss. Exposed collagen fibers from the damaged side will promote the platelets to adhere. When platelets adhere to the damaged vessel, they undergo degranulation or release cytoplasmic granules which contain serotonin, a vessel constrictor, ADP, and thromboxane, A2. The ADP attracts more platelets to the area, and the thromboxane, A2, promotes platelet aggregation, degranulation, and vasoconstriction. Thus, ADP and thromboxane, A2, promote more platelets adhesion and therefore more ADP and thromboxane. The positive feedback promotes the formation of a platelet plug. The final hemostatic mechanism is coagulation. The damaged tissue releases factor 3, which with aid of calcium ion will activate factor 7, thus initiating the extrinsic mechanism. The factor 12 from active platelets will activate factor 11, thus initiating the intrinsic mechanism. Both active factor 7 and active factor 11 will promote cascade reaction, eventually activating factor 10. The active factor, along with factor 3, factor 5, calcium ion and platelet thromboplastic factor PF3 will activate prothrombin activator. The prothrombin activator converts prothrombin to thrombin. The thrombin converts fibrinogen to fibrin. The fibrin initially form a loose mesh, but then factor 13 causes the formation of covalent crosslinks which convert fibrin to a dense aggregation of fibers. The platelets and the red blood cells get caught in these dense mesh fibers and thus form a blood clot.
Wow, that was interesting. But I have read that some members of the British royal family have problem in clotting of blood process.
Yes, hemophilia or bleeder disease is X-linked recessive genetic disorder in which the body does not produce the functional factor 8 essential for clotting. The females are the carriers and males express this trait. Hence, the disease can be fatal for men.
Have you heard of blood groups? It is the process of classification of blood on the basis of presence or absence of proteins on RBC's surface. Blood types differ from person to person. Among different blood grouping systems, two are the most important: ABO system and RH system. According to the ABO blood group system, 4 major types of blood groups are identified in humans. These are A, B, AB, and O. Karl Landsteiner first classified blood into blood groups A, B, and O. He named this the ABO blood grouping system. He observed that 2 distinct chemical molecules are present on the surface of red blood cells. ABO blood groups are based upon the presence of certain glycoproteins on the surface plasma membrane of red blood cells. These proteins are of 2 types, namely A and B, and are called antigens. There are 2 corresponding antibodies that react against these antigens and occur in blood plasma. Antibodies are named anti-A and anti-B. An antibody molecule reacts with a specific antigen and renders it highly sticky so that RBCs containing this antigen clump together.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of the cells, followed by complement-assisted lysis of these cells. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients. Type 2 hypersensitivities involve interactions of antibodies and surface antigens of the cells, followed by complement-assisted lysis of these cells. There are 4 different blood groups based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients. Type 2 hypersensitivities involve interactions of antibodies and surface antigens of the cells, followed by complement-assisted lysis of these cells. There are 4 different blood groups based on types of antigens.
Now we know that blood groups are differentiated on the basis of antigen protein present on the RBC surface. Membrane of each RBC contains millions of antigens. To know more about the ABO blood grouping, let us watch an animation.
Your blood group would have been surely asked if you have ever required a blood transfusion or donation. Have you ever thought, how did you come to know about your own blood group? How is your blood group different from your friend's blood group? Well, the blood is classified on the basis of different blood group systems. There are approximately 33 blood group systems, but the two most important ones are ABO and RH system. Let us understand an ABO blood grouping system. The type of blood group we possess depends on the presence or absence of antigens and antibodies in our blood. The red blood cells of our body contain specific antigens on their surface. The two main types of antigens are antigen A and antigen B, which determine our blood group. But how do we get 4 different types of blood groups from just 2 antigens? Let's find out the answer. Well, the different combinations of antibodies and antigens give 4 different types of blood groups. For example, if a person has antigen A on the red blood cells, his blood group is A with anti-B antibodies in the plasma. Similarly, if a person has antigen B on the red blood cells, his blood group is B with anti-A antibodies in the plasma. If a person has both A and B antigens on the red blood cells but no antibodies in the plasma, his blood group is AB. You must have heard that people with AB blood group are also called universal recipients. But do you know the reason? Well, the reason is quite obvious. People with AB blood group do not have any antibodies in the plasma. It means their blood does not discriminate against any other ABO type. Therefore, they can receive blood from people with any blood group, but can donate only to the people with AB blood group. On the other hand, if a person has neither antigen A nor antigen B on the red blood cells, but both anti-A and anti-B antibodies in the plasma, his blood group is O. Do you know people with blood group O are also called universal donors? Yes, you have guessed the correct reason. As we have already understood that people with blood group O do not have any antigens on their RBCs. Therefore, their blood will not be rejected when it is given to other ABO type. They can donate blood to any other blood group, but they can receive blood only from people with O blood group. Another important blood group system after ABO is RH system. It is named after rhesus monkey, since they were initially used in the research to make the anti-serum for typing blood samples. In RH blood group system, blood groups are classified according to the presence or absence of the RH antigen on the red blood cells. So, a person with RH positive blood group has RH antigens on their red blood cells, while a person with RH negative blood group lacks RH antigens on their red blood cells. To understand this, let's see some examples. If a person is RH negative and has blood type A, his blood group is A negative. Similarly, if a person is RH positive and has blood type B, his blood group is B positive. As each blood has specific antigens and antibodies, the knowledge of blood type is very important. If 2 different blood types are mixed, it can lead to clumping of blood cells, which can be potentially fatal. Therefore, before blood transfusion, a proper understanding of the blood group system, their clinical significance, and cross-matching tests are important.
Look at the pie chart to understand the distribution of the various blood groups in India. It's time to answer the correct. A person with blood group matching or compatibility. It involves testing blood from both donors and recipients. This ensures that a recipient is given blood that is compatible and is as safe as possible. If a unit of incompatible blood is transfused into recipient from a donor, a severe reaction may occur. Let us learn about blood transfusion. Blood transfusion is the transfer of blood from the donor to the recipient. Transfer of blood or its components into another person's bloodstream has been carried out for more than 100 years. Why is blood transfusion needed? It is needed to regain the blood lost due to excessive bleeding during surgery, certain severe diseases, or accident. Let us understand the procedure followed during transfusion. First, blood is taken from a healthy donor. After collection, matching is done between the donor and the recipient blood groups. Blood is also tested for any infection as it may cause harm to the recipient. Collected blood is then transferred to the recipient. Blood transfusion may lead to two possibilities. In case the recipient's body does not recognize donor RBC as its own, its result is fatal. What is the cause of fatal result during blood transfusion? When donor RBCs are introduced in recipient body, a reaction may occur between donor RBC antigen and recipient antibodies. This will result in clumping or sticking of RBCs together. This will lead to the blockage of blood vessels, thereby threatening recipient's life. Try to answer the correct RH factor is. Let us recapitulate what we have learnt in this module. Blood is a connective tissue that is made of a fluid matrix called plasma and formed elements. RBCs are non-nucleated cells and red in color due to the presence of the pigment called hemoglobin. WBCs protect the body against infectious diseases and foreign particles. Platelets are small, oval, rounded, or biconvex disc-like bodies with colorless and granular cytoplasm without nucleus. Antigen is a substance that evokes the immune response. Antibodies are specialized proteins that are used by immune system to identify and defend foreign particles to the body. 4 types of blood groups are A, B, AB, and O. Blood transfusion is the transfer of blood from donor to recipient. Rhesus factor is an inherited trait that refers to a specific protein found on the surface of red blood cells.